Funding for this podcast is provided in part by the IPM Network through the USDA National Institute of Food and Agriculture and by various commodity groups through checkoff dollars, including the United Soybean Board, National Corn Growers Association, and Cotton Incorporated.
The ICDeadPlants podcast shares the stories of people and plants, pests and pathogens, and the conflicts among them. Join us as we speak to the folks who attempt to solve complex problems with plants through pest management research. ICDeadPlants is an extension-based podcast created by the Crop Protection Network and the North Central Integrated Pest Management Center and hosted by Ed Zaworski.
Welcome back to the ICDeadPlants podcast. As always, I'm your host, Ed Zaworski. Today, I am joined by Danielle Mikolajewski, who is currently at the University of Florida. But we're going to be talking about chestnut blight and a phenomenon that I know nothing about within the field of plant pathology, which is hypovirulence. So just a little bit of background on Danielle. So she got her bachelor's at the University of Delaware in plant sciences, followed by her master's also at the University of Delaware in urban forestry / soil science, and then Ph.D. at West Virginia in forest sciences and biology, where she studied Dutch elm disease and chestnut blight, right?
Dutch elm disease was my master's.
Dutch elm
and
disease
chestnut blight was the PhD. Mm-hmm.
Ah, gotcha. And then currently, I'm going to kick it to you. What are you doing currently, Danielle?
So currently, I am a postdoc at the University of Florida Institute of Food and Agricultural Sciences, Fort Lauderdale Research and Education Center.
Thank you.
So,
and what do you do there?
IFAS FL Rec is the acronym. And I am studying palm disease rots. Gotcha.
Today, the topic of the hour is chestnut blight. But before we get there, Danielle, I like to get to know you a little bit, get the audience to get to know you. How did you end up in the wild field of plant pathology? What was your path to get there?
So in high school, I originally wanted to be a dentist, soon to realize that there's blood when you do any kind of human doctoral profession. And I'm okay with my blood, but other people's blood, once it gets to be too much, is a little much for me. So I shifted gears. I really enjoyed the environment and I enjoyed math, funny enough. So everyone told me that I would be good at engineering. So I started thinking that I would do environmental engineering. But I came across the Future Farmers of America chapter of my high school, so FFA, for those of you who don't know, because actually Future Farmers of America is done with. It's just FFA now, unless they changed it again, but it should just be FFA now. And I took a plant science course, and I really enjoyed it, and I won first place in the floriculture competition back at my freshman year of high school, and I kind of stuck through all of that. Soon, it was time to start looking at colleges, and I knew I wanted to go to the University of Delaware because that's where I'm from. I was right up the road. So I really didn't think about going anywhere else for college. I went in towards the engineering department, still thinking I wanted to do environmental engineering. And he starts explaining, the guy starts explaining what they do, and it really kind of seemed like they built HVAC systems. I'm sure I'm getting that wrong for any environmental engineer out there.
Makes sense.
But that's kind of what he was describing to me. And I kind of paused and I was like, wait, don't you guys do research and like play with plants and stuff? And he was like, oh no, you either want environmental sciences or plant sciences. And again, having an agriculture background from FFA, I decided I would check out the agricultural college first. And I just fell in love with the landscape there and the people that I met in walking through the greenhouse. And I was like, yeah, this is where I want to be. So I applied to the University of Delaware under plant sciences, and that's what I did. And I actually, through my college career, also got a double major in agriculture and 2 minors in horticulture and soil sciences. So from there, knowing that I was going to go to the University of Delaware, I started looking for a part-time job. And my mom actually was in contact with someone who knew someone at the university, and they had heard that a professor was looking for an undergrad to just kind of help out in the lab. So actually on my graduation day from high school, I went and interviewed with Dr. Jung-Yoon Lee at the University of Delaware and got the job as an undergraduate assistant. And I think as a lot of people starting out, I washed many dishes.
Yep.
I mean, I think because they did a lot of transformation work there, I was washing dishes like 2 to 3 hours a day, I think. But then I got to do some cooler stuff, like helping out with some of the transformations, and started working with more of the plants, but sadly lost funding to keep me on. So I went back to the university and I talked with Dr. Sue Barton, who she ran a program at the university. I don't remember what all the acronyms were because I didn't actually do the program, but she basically ran an internship program for the university. So I figured she'd have some ideas on where I could find a new job. And she's naming all kinds of stuff. And again, I was like, do you guys do any lab work anywhere? Like I love the outdoors and whatnot, but I was like, I don't want to be planting plants with my degree. I was like, I want to do research. I want to find things that have not been discovered before or expand upon what has been discovered. And she put me in the direction of my academic advisor, Dr. Nicole D'Onofrio. I interviewed with her and she was like, do you like fungus? And I was like, I don't know. We'll find out. I was like, does it smell? Because that was my big thing. If it doesn't smell, I'll be fine. And I think I set my hand on fire like one of the first days in the lab. And from there, just fell in love with plant pathology. So in her lab, I worked on Magnaportha oryzae, which is rice blast.
Mm-hmm.
And again, we did a lot of fungal transformations and screening to look for new genes. And I believe what came of that was that my Ph.D. student that I worked for, Dr. Jess Cooper Pancake, which is an awesome last name, she discovered a new YAP gene with the research that I helped her with. So then from there, I wasn't quite ready to move out of Delaware. And I was still interested in trees. I always said I was going to save the trees. So as much as I love doing research, I wanted to get out into the forest a little bit. So Dr. Tara Tremmel was fairly new, but she worked on urban forestry. And she had a project that she just got funding for on Dutch elm disease. So she didn't really have the plant pathology background, but I didn't really have the urban forestry background. So I applied and got a master's position with her working on Dutch elm disease. So I didn't actually get to work with the fungus, but what I did do was plant 90 possibly resistant clonal varieties of American elm in Newark, Delaware, and Philadelphia, Pennsylvania. And then there was another set that was planted in Columbus, Ohio. I kind of shifted, did a little bit more urban forestry stuff, but I really missed the plant pathology. And I remembered Dr. Matt Caston from my times as an undergrad at the American Phytopathological Society meetings. And I remember trying to pay attention, but as an undergrad, I don't know if others feel the same way. You don't know what the heck is going on. Everyone's throwing these terms at you and, you're trying to pay attention, but some of it gets a little boring if you're not interested. And I remember him getting up on stage to give his talk and he just excited the crowd. He was so animated and I am also very animated. And I just remembered his talk on Chestnut Blight. And so I shot him an email. I was doing a master's during COVID, so I had no idea who would have funding to bring people on. But I shot him an email out of the blue and said, hey, I remember, you from APS. Would you be interested in bringing on a PhD student? We talked and he basically told me that I would be hired as soon as I applied. So nice. I applied, got hired. And that's where I truly found my passion for forest pathology.
In Chestnut Blight with Matt.
Mm-hmm.
That's awesome. Yeah, I did a podcast, feels like 10 years ago at this point with Matt. We were jumping on the bandwagon when the show The Last of Us came out. And Matt had just done like a magazine article interview with Barstool or somebody and asking just general questions about it. And somebody threw out to me like, hey, Matt would be a great guest to have somebody on as a mycologist to talk about this stuff and cordyceps and all that stuff. So yeah, I totally see why you gravitated towards Matt. He's a
yeah,
dude.
he was a wonderful advisor, and we're still really good friends. Again, we both bounce off of each other's ADHD. To just come up with the craziest ideas and get so animated and excited about these topics. Yes, he's done many interviews about The Last of Us, and people always ask, like, is this possible in the future? And he's like, not for hundreds of thousands of years, probably.
If at all.
If at all. Yeah, exactly. If at all. But yeah, between being a mycologist, he's also an entomopathologist, I guess is how I would say it. And then he's also a plant pathologist. So he does a little bit of everything.
So you got some good exposure there to some different stuff.
I did indeed. I am definitely not as skilled as some of my other lab mates were at like IDing mushrooms, and I still am not the biggest fan of insects, but actually coming here to Florida, I'm now turning into the insect mushroom person because I at least know more than the people do here.
Want to keep up with the latest integrated pest management research? Check out the North Central IPM Center's Pests in Progress webinar. Listen real time or check out the expanding library of past recorded webinars and keep up to speed.
So, chestnut blight. I guess before we get into first of all, I didn't even know how much research was still going on in the area of chestnut blight. I think generally speaking, as a plant pathologist studying plant pathology as for my master's, I generally knew about chestnut blight as this historical pathogen, historically significant in that it came in. It was a introduced pathogen to the United States, and it came in and wiped out the American chestnut. Kind of one of the few things that gets held up is like, all right, this was a huge problem. Well, up there with like the late blight of potatoes or something, this huge pathogen coming in and doing a lot of damage. But I guess I was really unaware of what is still going on today, and that's eventually what we're going to talk about. But before we get there, I kind of want to talk about just general basic stuff for our listener. If they don't know anything about chestnut blight, let's take them through that. So what is chestnut blight, Danielle?
So chestnut blight is a plant disease that is caused by a non-native fungus. Its name is Cryphonectria parasitica, and it was first discovered in America in the New York Zoological Garden, Botanic Garden, in 1904. It was delivered, I think we've decided, from China through genomics. We've decided that it came through China on some infected wood that was imported and landed in New York. It also was discovered a little bit later in Europe on European sweet chestnut. It causes the same symptoms in both places. The fungus itself is orange, which is very morphologically characteristic of it, so it's easy to identify, though a lot of members in that genera and family complex can also be orange, so sometimes you still need genomics to figure out which one you've got. It is a canker pathogen. So what it does is it clogs the xylem vessels and then starts eating at them. As a defense response, the plant or the tree starts to form a canker, expanding its wood to try and wall off the pathogen. But this does not work. But it causes this canker, which the fungi then protrude out of and form these little orange bundles of tissue called stromata, which eventually the spores are produced out of. So clogging the xylem and killing it, everything above that canker now cannot receive any nutrients from the root system. So everything above that canker, if aggressive enough of a pathogen, will just die. So originally you'll start seeing some wilt symptoms, but eventually the top of that tree will just die and fall off. As the spores are released, they will continue to reinfect the original tree that it was growing on and will also infect surrounding trees. So eventually this pathogen will just kill the whole trunk of the tree. However, it does not kill the root system. So the chestnut, the American chestnut, or sweet chestnut will start to sprout from the stumps, and you'll basically just see these branches popping out. But these will also eventually succumb to the blight.
Hmm.
So really, when you go out into a forest, the chestnuts that you'll see are usually these shrubby stump sprouts, and they're usually covered in the orange stomata.
Gotcha. So that's kind of the current status right now, is that rather than the glorious American chestnut of old, we've got shrubs that are kind of on a limited time until they themselves succumb to chestnut blight. They're kind of just keep proliferating like that basically at this point, right?
Mm-hmm.
Yeah, with the canker diseases, I always tell if I'm doing extension talks, and in Iowa, the most common thing I'm talking about is spruce trees. There's lots of spruce trees and people's windbreaks, and they can get a disease called cytospora canker. And I always like to tell them it's like a tourniquet, except it has the opposite effect. You're trying to stop toxin or something coming through your arm, and you're tying that tourniquet on there. It's the opposite. The canker's acting as a tourniquet, and it's cutting off that extremity, so it's killing it. And in the case of this, the whole trunk of the tree, killing the whole tree, but not the roots. Kind of working back really quick before we get into it. I mean, it was identified in 1904 in the New York Botanical Garden. What happened after that? Just a brief summary, like.
This pathogen started spreading, and it spread quickly. It spread 50 miles per year is what they were calculating.
Wow.
And the native range of American chestnut is Maine to Mississippi. And it can still be found even in some other southern states like Florida, Georgia, and then all the way up into Canada as well. So it started in New York, and it just started spreading. Eventually, by the 1940s, 1950s, it wiped out 4 billion trees, which was 90% of the American chestnut population, which at the time was like a good 25% of our forests, because American chestnut was the dominant forest species in its native range, especially in Appalachia.
Yeah, I've seen pictures, and the size of the trees is one thing, but then to think that it's they made up almost a quarter of the tree population in some places, to see that leave the landscape must have been a incredibly powerful or striking or frightening or whatever adjective you want to use thing for the people in those areas. So essentially, current day, I think I Googled what is the status of the American chestnut, and it said not extinct, but not out there in the landscape really doing much. Fast forward to the current day, what types of things are being done to either try and maintain the chestnut, revive the chestnut? What types of research is being done out there?
So originally, scientists tried to use injections, fungicides, but none of those worked. So we have no chemical control methods currently for chestnut blight. The 3 things that we do have that we are currently working on are hybridization, back cross breeding, transgenic breeding, and something called hypovirulence, which is what I study.
And so, I think we can all maybe imagine, I'll give my best summation, and Danielle can tell me if I'm wrong. So we've got just crossbreeding between different types of chestnut throughout the globe. Some that have, evolved resistance or tolerance to chestnut blight, trying to work that way in. Transgenics being what you're trying to either with, well, I guess I don't know. I mean, is it traditional transgenics? Are people using CRISPR now to try and insert genes into? Danielle, are you aware?
They most certainly could,
okay.
But the basic one that they've been using is Agrobacterium.
Okay. So, and then hypovirulence, which is what I really wanted to talk to Danielle. Obviously, chestnut blight is a hugely important disease historically for anyone studying plant pathology. So it's one that we can all kind of understand, at least at its basic level. But I had never heard of hypovirulence, and I guess I can't speak for my audience. Who knows? Maybe I'm the odd man out here. But Danielle, what is hypovirulence?
So in the simplest terms, a hypovirus, which is the same thing as a mycovirus, so fungi infecting virus, invades the fungal tissue and lives in the cytoplasm and causes the fungus to elicit less severe symptoms on the tree, basically weakening it. Thus, we see these trees starting to, quote unquote, heal themselves. And that's kind of the most basic description I can give you.
So basically, the fungus itself, Cryphonectria, is getting infected by a virus. So a pathogen is being infected by a pathogen. It's basically making that pathogen less virulent so the tree can kind of start to come back. It can start to heal over that with that callus tissue that it's producing over that canker and kind of stop the bleeding.
Yeah. And so this was discovered in Europe, specifically Italy, I believe, where some scientists went out to do some tests and they realized that some of the trees were starting to heal. And so they took some samples and discovered this virus. So there are 4, we'll call them strains of the virus. And again, sorry, virologists. I know I am not a virologist. I am a mycologist. So I don't know the full naming system here, but I've been calling them strains. So they're CHV1 through 4. And CHV stands for Chestnut Blight hypovirus. And then there's 1, 3, and 4. 1 was discovered in Europe, and that is the one that is most commonly used. It was imported into the US and is used as a biocontrol agent. It causes a white phenotype in the fungus. So remember before I said the fungus is orange.
Oh.
So we can ID it pretty easily if the hypovirulence is working because we're going to see white and not orange. But then we also have a few native strains 2 through 4 here in the United States. 2, 3, and 4 cause less severe symptoms and aren't as virulent attenuating. So they don't cause as much harm, and they can sometimes go unnoticed because they do not change the color.
Oh, interesting.
One of them changes the morphology a little bit. The fungus itself usually grows pretty uniformly in a petri dish and pretty fluffy, I would say. There's some aerial hyphae where CHV2 produces a brown phenotype and was discovered in New Jersey. CHV3, which also goes by GH2, was discovered in Michigan, and that gives these lobed margins.
Oh, interesting.
So no aerial hyphae, and they look kind of like the fungus is growing on top of itself. It's very thick. And then CHV4 really causes no outward symptoms on the fungus and is found in Appalachia.
Gotcha.
So we have our own native strains of the virus, but we tend to use the one from Europe because it causes the most harm and it causes that white phenotype, which is easier to identify.
Causes the most harm to the fungus, right?
Yes, to the fungus, to the fungus. Thus,
the
tree has a better chance of survival.
Gotcha.
And then there's different strains of the strains too. So that's why I'm I don't know the terminology fully.
Sure. We got the CHV 1 through 4.
Yes,
we'll keep it at that 4. Level. So I'm just kind of curious, Danielle. I mean, along the lines of hypoviruses, are there? You can feel free to veto this one if you don't know, but I'm just kind of curious. Are there any other instances in plant pathology where we see hypoviruses come into play? Are there any I don't know, historically significant ones or anything? Or maybe we don't know. I don't know, but yeah.
There are definitely some that have been reported. I read some papers, again, would have to go through my literature review to figure out which ones, but there were definitely some that have been reported. Because they're not my specialty, I don't know how helpful they are as a biocontrol and if they're super important and used a ton or if they were just kind of found.
Okay, so suffice to say there have been other documented events where there's been hypovirulence present in a pathogen. I guess my next question about this is, okay, we know that these hypoviruses exist, and we know that they can kind of knock back that pathogen, the Cryphonectria. How are they deployed? How does one get the virus to the fungus?
So this is the fun stuff that I was working on. The best way to explain how this fungus works is to use an analogy of blood types.
Okay.
Let's just pretend I don't even think I know my blood type. I think I'm like A
Oh,
something.
A.
Yeah, so I'm like, let's pretend I'm A positive. I don't know if that's a possibility. I'm A positive.
Yeah.
If I'm in a car accident or I'm shot or whatever, I need a blood transfusion. I need to have A positive blood given to me.
Mm-hmm.
Or else my body is going to reject it. I go into like anaphylactic shock or whatever. My doctor boyfriend's going to be so mad if that's wrong. And I die. The same thing happens with this fungus. There are 6 genes. They are called VIC genes, which stands for vegetative incompatibility genes. You might also see them as VC or VCG, depending on how old the paper is or what pathosystem you're working on.
Sure.
But most fungi have this system. And it is a non-self recognition system.
Ah, okay.
So that the fungus doesn't allow anything inside of it that is not itself.
Right. Thus the blood type analogy, right?
Yes, thus the blood type analogy. So for Cryphonectria, there are 6 of these genes, and they are diallelic. So there's 2 possibilities at each VIC gene. That gives us a possible combination of 64 strains of chestnut blight that are out there.
Okay.
If even one of those VIC genes differs in allele, so let's say the way we denote it would be like 1111-11. Let's say that's one VIC genotype in Cryphonectria. If there's a 2 at any of those spots in another strain of Cryphonectria, the fungi do not combine. And the term we use is anastomosis. So that is the combination of the fungi by creating hyphal tubes that allow for the exchange of materials. And this could be nuclei, nutrients, cytoplasm, or in my case, the hypovirus.
Gotcha.
So that virus will not spread if the fungi don't match. In Europe, this is not as big of a problem because they have not had as many introductions to the fungus. So I think one of my last counts from the literature, they have like 30-something VIC genotypes that have been discovered.
Okay.
Here in America, we have found all 64.
Ah.
So,
of course.
Because we've had more, I know, of course, because we've had more introductions of the fungus to different locations. So even though it was found in New York, it came in after that as well in different locations. And it diversified as well. So the spread of hypovirulence in Europe works decently.
Okay.
Because there is less VIC gene diversity. In America, it doesn't work as well because we have high VIC gene diversity. Usually what you do is you take a survey, this is what you should do. What people used to do was just put hypovirulent strains of chestnut of Cryphonectria out there and hope for the best.
Right.
What you should do is sample different areas, again, depending on maybe how big your area or state is. Maybe you sample 50 trees in one spot, 50 trees in another spot, sample multiple cankers on a single tree, however you decide you need to do it to get the best picture of the diversity of the fungus in your area. Bring them back to the lab, VIC genotype them, which you can either do by pairings, but previous work in my lab, in the Casson lab, developed primers. So you can just sequence and figure out the genes from there.
Gotcha.
It still takes a while, but it's easier and takes less time than if you were to take one strain and pair it with every single 64 possible combinations.
Sure.
You then will make hypovirulent forms. You get the virus in there, injection or whatever. I mean, like not with like a needle or anything, but like, technology into one strain and then get a matching strain, plate them together. Hypovirulence occurs, and actually on the plate, you'll see that change from orange to white.
Oh.
Take a plug of that white, and you can keep culturing that. You don't have to keep putting the hypovirus back in. That hypovirus will proliferate, and you can just get multiple cultures. And then you will take your hypovirulent strains and put them out in the forest.
Okay.
Usually we use a punch, like a leather punch, to make a hole in the tree. This fungus does need to be physically inoculated. We can't just like paint it on a canker. Previous work in my lab showed that. And you stick it in, fungicide down, cover it up, and hope it proliferates. there's many complications with this because it's a very complex system. But
Sure.
you may transfect a lot of the strains that are in a single canker
Okay.
using like a slurry of the most common Vic genotypes you found, but you're going to still have some left that didn't have that same Vic genotype, or the fungi is going to eventually diversify its Vic genes and change. You're going to be left with some strains that did not get the virus.
Sure.
And then they are going to proliferate and outcompete the virus strains, the strains with the virus,
Right.
and then take over the canker and keep going.
Gotcha. So you're putting selection pressure on the ones that...
Yes. I think that's what I was thinking of, selection pressure.
Gotcha.
What I was working on, and what previous members in my lab, and I believe at the University of Maryland is where they were located, developed a system to hopefully bypass this Vic gene need. And we call it the super donor because it is super and donates virus. And going back to the blood type example, O
yeah.
blood
Okay. Yeah.
is the universal donor. You can think of the super donor like O blood.
Got it.
So what they did at the University of Maryland, they sequentially knocked out the Vic genes.
Okay.
So that they were left with strains of the super donor that didn't have all of those Vic gene barriers. They did not knock out Vic4 because it doesn't have anything to do with virus spread. And they couldn't knock out Vic2 because that one is needed for fungal survival. If you knock out Vic2, the fungus dies. So these strains of the super donor have an allele of Vic2 and Vic4.
Got it.
The rest are knocked out. Previous work in their lab, previous work in my lab showed that these work way better at disseminating virus than just putting hypovirulent strains out there or, a control.
Gotcha.
So I was brought in to find any limitations to this super donor. Because as you go through a regulation process, eventually the goal is that this would be a biocontrol agent that is regulated by, the government.
Yep.
That could be used to manage chestnut blight.
Mm-hmm.
I was brought in to find any loopholes to that.
Sure.
Is there anything that the super donor could do that could be harmful? Are there any situations where it doesn't work?
Mm-hmm.
And we found some strains from Europe of Cryphonectria parasitica. They're called 65 through 73.
Okay.
That have the same Vic genotypes as 1 through 64. But for some reason or another, previous literature said the super donor would not work.
Interesting.
Or they predicted that the super donor would not work. So I tested the super donor against those, 8 strains of Cryphonectria. I found that it worked on some, but it didn't work on others. And we did not get to delve deeper into that and to why that's not happening.
Sure.
We have thoughts, hypotheses that could be tested. We have the thought that some of these strains of Cryphonectria have evolved a new Vic gene. Because if it's got a new Vic gene and the super donor doesn't have that Vic gene or that Vic gene knocked out, then it's not going to match up and it could stop that process of the virus transfection.
Gotcha.
We've thought that there could be different alleles. Originally, we thought maybe there could be other hypoviruses, but our virologist said that that's not likely because CHV1 is so debilitating and can outcompete for space. Basically, we want to take those strains that super donor didn't work on and sequence them, like full genome sequence them, take apart the whole genome and see if we can find anything new that could be stopping the super donor.
Gotcha.
Again, that didn't get to happen, maybe in the future, hopefully. But that's what I did. I was developing this biocontrol agent to use against chestnut blight.
Are you interested in reading the latest integrated pest management updates such as current pest concerns, management tips, research updates, available IPM resources, upcoming events, and job announcements? Make sure you subscribe to the monthly newsletter, The Central Issue, published by the North Central IPM Center.
Makes sense. And the concept of the super donor also makes a lot of sense. I was kind of chuckling in my head because I was already like gearing up to ask you a question like, so do you guys have like essentially donor fungi of all of those 64 different Vic gene combinations of the Cryphonectria? And then you answered the question already with the fact that, all right, no, this makes much more sense, is that you have one super donor that could potentially perform anastomosis with any of those and transfer the virus. I'm curious, one other question I was kind of holding on to, Danielle. I mean, how specific are these hypoviruses like the CHV1 through 4 to Cryphonectria? Are they closely evolved with that pathogen? Is there any possibility of them, I don't know, harming beneficial fungi in the environment? Do we know? Is that part of what needs to be, what you were researching?
That's a great question. I did some work on that.
Mm-hmm.
One of my projects was looking at exactly that, but we took closely related fungi. So I found some Cytospora and I found Endothea gyrosa, which has been misidentified as Cryphonectria parasitica. In fact, the whole naming system when I was going through the literature for my dissertation was a mess. So Endothea gyrosa is a weak pathogen on Crocus species and other species in the Fagaceae. And so one thing I tested was to see if it was more virulent than parasitica. We got results that basically said, for the most part, no. Both of them are kind of weak on other Fagaceae hosts. Though we did have significance with Cryphonectria parasitica on red oak for one experiment. And I believe Matt Casson and Amy Matheny, who I want to mention for sure, who helped me with a lot of this stuff, are going to start some new pathogenicity assays out in the field. One of my other experiments was looking at hypovirus transfection using the super donor to Endothea. Because in theory, and actually I did a little bit of work on this. It wasn't finished. So many projects I didn't get to finish. Endothea does have Vic genes. I think I found Vic 1, 2, and 3. I don't know if it has 4, 6, and 7. So in theory, Cryphonectria, if it was infected with the hypovirus, it could transfer virus to Endothea. In theory.
Gotcha.
I started doing some stuff in the lab. Using the super donor, it worked. I was able to use super donor to donate virus to Endothea strains, Endothea gyrosa strains. I did not get Cryphonectria donating it to Endothea and vice versa.
Gotcha.
So in the field, it is very unlikely that this pathogen, that this virus, though it can infect other Cryphonectriaceae genera
gotcha.
and species, that it would happen. However, with the super donor, if we get to a point of regulating it and releasing it, if Endothea is also out there, I don't know what the likelihood is of it transferring virus.
Gotcha.
Now, I don't think this would be a problem because Endothea is another pathogen, but you never know. You got to do all the testing and make sure, like I said before, you got to do all kinds of testing to make sure that nothing's going to harm the environment or anything. To answer your question in more simple terms, no, the virus is not going to harm any beneficial fungi or any other fungi that are outside of the Cryphonectriaceae.
Gotcha.
They wouldn't do that, but if they harmed ones within the Cryphonectriaceae, we probably need the super donor to donate virus. It won't just happen naturally.
Just to follow up on that and make sure that I understand as well as the listeners. So let's say you've deployed, and folks, this is hypothetical. So let's say you've deployed the super donor. And so you've cut a plug out of a tree. You've deployed the super donor into that tree. It infects the different strains of Cryphonectria within there, or with the different Vic genes. It's successful. Everything's good. Do you then, if you were to go around and you were wanting to just cure as much of the chestnut blight in the landscape as you can, do you have to go around from tree to tree? Does the fungus itself now disseminate the virus from tree to tree via spores? Like, what's what's the next steps of that?
So that was going to be a project that I was going to work on. But from what we know from previous experiments, that the super donor was only disseminated once and, going back out into the forest and looking and seeing what happened. After a year, the super donor does break down.
Gotcha.
So you can't just apply it once. We don't know how often we need to apply it, but we know it's not just like a once a year thing.
Gotcha.
Studies have shown that the virus can spread through spores.
Gotcha.
However, part of the attenuated virulence and making it less aggressive, the fungus does not produce as many spores.
Gotcha.
So
yeah, that makes sense.
The virus does transfect spores at a specific rate, but so many folds spores are made versus the wild type. So it's not being disseminated as often because not as many spores are being made. So again, concluding that question would be, we know we need to apply it more than once. We just don't know how many times a year.
Yep, makes sense. You're knocking back its reproductive fitness, so you probably can't expect it to do the job for you. So
exactly. And that's why some people, we've talked about using our more native strains because they do not
interesting. Yeah.
lessen spore production as much as the CHV1.
Yeah, I had kind of forgot about that whole.
But then they also don't hurt the fungus as much. So it's like,
Man, yeah. So there's a lot of avenues there to potentially tease apart in the future.
Exactly.
Well, Danielle, at this point, before I get into wrap-up questions, so I've got some questions to ask you on the back end. Did we glaze over anything as far as you can tell as about Chestnut Blight or hypoviruses or anything?
Should I explain why American Chestnut is important?
Yeah. Well, yeah. I think. Well, yeah. Go ahead.
Okay.
Well, here. How about this? Rather than explain, because you could go down a whole rabbit hole there, and you could get into like historical significance. How about why is it important to this? This might eat into my wrap-up questions, but I'm willing to risk it. Yeah. Why is it important to do this work to try and bring back the American chestnut?
So American chestnut is a historically significant tree. It has many, many uses. So I'll just name them kind of without going into much detail. They have tannic acids inside their wood, which makes the wood very rot resistant. So it's very good for building materials. A lot of stuff used to be built out of chestnut wood. And those tannic acids can also be used in leather working. It is a very large, fast-growing tree. So it makes for good recreation purposes, such in like a park or at a university, but is also an urban tree that could help with climate cooling. So it could be planted in urban settings to help curb the impacts of our roadways and sidewalks. And then its nuts are not only used for food for us, but also for wildlife and livestock. Once American chestnuts started to disappear, people tried to replace it with other trees. Specifically in Appalachia, what took over was oaks, maples, and hickories. But the nuts, they just didn't do what the chestnuts did. They were just not as rot resistant in the wood. They have other purposes, don't get me wrong, but they just couldn't replace the American chestnut. So the reason we really advocate for this work and trying to bring American chestnut back is because of not only the historical significance, a lot of people just want to see it back, but also because there are so many uses for it. And it should, in theory, grow fast enough that we would actually be able to use it within 10 years of its growth.
Oh, wow.
Versus like oaks that like take forever to grow. Makes
sense. There's huge, suffice to say, there would be huge socio-economical implications of being able to bring the chestnut back.
Do you need to keep up to date on new pests or pests that may be moving into your area? Pest Alerts focus on new pests, pathogens, and weeds that are popping up in the North Central region. Check out the North Central IPM Center's Pest Alerts page and stay a step ahead.
So that is a perfect segue into my first wrap-up question. This is going to be speculation on your part, I know, but what do you see as, like, what does this research mean and these developments with learning more about the pathogen? What does it mean for the future of American chestnut as you see it?
I want to say that as much as I love my research and I want to continue doing my research, it's very important to collaborate with the other research that is going on with the transgenic trees and the hybrid trees. So I think, for starters, we need to employ all three management strategies at once and not just one at a time. Now, there's still plenty of work that needs to get done behind the scenes before we can get there. But I think eventually we need to come up with management strategies using all three of these management practices. I think we definitely have way more research that we need to do. Maybe not in our lifetime, but hopefully our kids and grandkids will see this research come to fruition and we get American chestnuts back. But along with these three management strategies, we'll also have to employ other management strategies that are not pathogen related. So, we have to
just
figure
like
out
forestry
what
management.
the planning time is. How much, do we need to water them? Do they need fences so that deer don't get at them? Like, how long do we have to do this? I mean, do we only have to do this for a year? Do they need it for a really long time? So there's different avenues that we're going to have to explore once we actually start getting into the reforestation process, to figure out how to establish these trees so that they don't just die as soon as they go in the ground. We're definitely going to need to help them along eventually. And this might be like, we're talking like great grandkids, maybe. Hopefully they'll be able to survive on their own again and take over where they used to. But I think there's definitely some different management strategies we're going to have to do to get them to that point.
Given the economic importance, so you mentioned the rot-resistant wood, the masts, do you envision people kind of growing American chestnut in a kind of commercial fashion, like similarly to what you see with like southern yellow pine as an example? I don't know. I'm just curious. Yeah,
I do. There are already chestnut orchards around. A lot of them are probably growing non-native chestnuts, like Chinese and Japanese chestnut.
Gotcha.
To make up for our loss of American chestnut. But with the hybrids and the transgenics, some of them have started to be disseminated to specific growers who we can count on to manage them properly and give us feedback on how they're doing in their orchards. Some of them are being planted in monoculture. Some of them are being kind of integrated in with other chestnut trees. But yeah, I believe that there will be eventual mass production in orchard settings.
Gotcha. So they're almost out there now as in a sentinel function, like they can see how successful they're going to be, but not moved towards mass plantings or anything like that.
Yeah, not yet.
Gotcha. My next question for you, Danielle, is kind of about hypovirulence. I hear about, I like I said, I didn't know about this, but when I hear about something like this, and it's from a very naive perspective of I don't know a lot about it other than what I read leading up to this podcast. But in your perspective, something like hypovirulence, do you see this as like being able to contribute to other pathosystems? Like, do you think that there are a lot of other hypoviruses specific to different plant pathogens out there that might be utilized? Or do you think this is kind of like a one-off or random thing that we were able to find and maybe there aren't that many more? I don't know. What do you think?
I definitely think there are more out there. I think it's going to get narrowed down to how useful are they? As I said before, there's literature out there. Hypoviruses have been found in other pathosystems.
Sure.
I just don't know how debilitating they are to the pathogen. There might be some already out there, or we might be able to find more. Again, it's going out there into the fields and looking at whatever plant you have and saying, hey, this one looks a lot better than the others. I wonder, it could be an escape plant. It could have resistance genes, or maybe there's a hypovirus that's infecting whatever pathogen you've got going on. There's also, again, going back to some of the transgenic things, maybe there's a way to kind of like up the virus and make it a little more virulent
right.
the pathogen, but you got to be careful with that too, because we don't want to create a super pathogen either. But there's possibilities of that, maybe, of looking through the genome of these different hypoviruses, seeing which genes they use for pathogenicity of the pathogen,
Sure.
of the other organism, and then possibly create some new transgenic biocontrols from that.
Yeah, it jumped out at me right away as something like, man, another, because I'm constantly, I work with the integrated pest management team here at Iowa State. So I am constantly thinking about like, all right, it's all over the news, chemical resistance, pathogens resistance to chemicals is popping up more and more. So the idea of IPM and integrating more types of control into a control regimen for any disease is very intriguing, and this one is intriguing to me as well. So,
and you hit the nail on the head. I want to emphasize that we can't just use one control method. So like, even if you have resistant plants, you can't just plant them in monoculture. We need to continue to use a regimen of chemicals, resistant plants, biocontrols, cultural practices. You can't just use one thing, or else the pathogen is going to evolve and take over once again.
All right, Danielle, it's time for the hard one at the end. Well, it's fun, but it's hard. If you had to sum up your research specifically with hypoviruses and chestnut blight to a member of the public, and, give it to them in a succinct way that was impactful, what would you say to them?
See, the problem is, so I've actually, I won the 3-minute thesis talking about some of this research. So, I can do this. It's just so difficult to get it in such a small package, I think, for this, because there's so much you have to explain. There's 100 years of research. There's the Vic genes, the hypovirulence, the super donors. So, it's just so much to talk about. So I'll do my best, but it might be a little longer than some of your other guests.
Give it to me as if you're talking to somebody at Thanksgiving.
Yeah. So, okay. So chestnut blight is a non-native fungus that has wiped out 90% of our American chestnut trees. One method that we are developing to use against this pathogen is called hypovirulence, where there is a virus that infects this fungus so that the fungus is not as harmful and the tree can heal itself. The problem with this is that there is a lot of genetic diversity in the fungus, and thus the virus does not spread easily. So we're developing a biocontrol agent to bypass this genetic diversity so that it can spread the virus without the need of some of these important genes.
Applause, everyone.
Thank you. I would love to go into more detail, but that's the sum of it.
It's so hard to do because you want to go into more detail, but that was a great summation and a great thing to leave off on. So at this point, Danielle, it's your opportunity to make any acknowledgments, point people to any resources about chestnut blight or hypovirulence. You've got the floor for as long as you need it. Go right ahead.
Thank you, Ed. So first, I want to definitely thank the Casson Lab. Matt Casson, Amy Matheny, you guys are awesome. You know way more than I do about all of this. I'm so glad to have the opportunity to have worked with you, and I will definitely be collaborating with you in the future on more chestnut stuff. I would also like to finally acknowledge my undergrad assistant, who is now doing a master's at the University of Georgia, Emily Holloman. I would not have been able to do all of this without her. She helped me so much in the field and in the lab, prepping things, culturing, whether being side by side or on her own. So I'm really proud of her for that. And I want to just say thank you for her. If you guys would like any more information, you can contact Dr. Matt Casson or Amy Matheny by looking at the WVU website for their emails, or you can always reach out to me and I can give it to you. And you can also contact your local American Chestnut Foundation chapter. There should be at least one in each state, and you can learn what you can do to help. Friends, family are always helpful and wonderful. And then I'll just plug that I am now a postdoc at the University of Florida, and I'm working on palm diseases. So if you are curious or have any interest in palm trees, or maybe are having issues with your palm trees, you can contact me, or one of my two advisors, Dr. Brom Dillon and Dr. Micah McMillan.
Thanks again, Danielle. Folks, thanks for listening. Keep listening. We'll see you next time.
ICDeadPlants bridges the gap between plant science research and the impact it has on our daily lives. It is brought to you by the Crop Protection Network and the North Central Integrated Pest Management Center, which are both partially supported by the USDA National Institute of Food and Agriculture. For more information on today's topic or other pest management issues, check out our show notes, cropprotectionnetwork.org or ncipmc.org. Remember, this information is for entertainment purposes only. The statements made in this podcast should be interpreted within the limited context of the particular topics being discussed. Contact your state extension program for local information on pest management. For the full non-discrimination statement or for accommodation inquiries, go to cropprotectionnetwork.org/about. Music written and recorded by fellow plant pathologist Sean Toporek. Thank you for listening to ICDeadPlants.